This is Chapter 3, Part 3, Cells and Living Units, the Nucleus, Transcription and Translation. In this chapter, part of the chapter, we're going to focus in on the nucleus, which we refer to as the control center of the cell. This contains the nuclear envelope or nuclear membrane, the nucleoli or nucleolus, the chromatin, and specific protein sets that we'll see throughout the nucleus. This would be the area of the nucleus. Here we see the nucleolus, and then the nuclear envelope, or is another term for the nuclear membrane. The reason we refer to this as the control center is this is really our genetic library. Here we have, within the chromatin, we have the DNA, and the DNA acts as blueprints for nearly all of the cellular proteins that we'll see being important for how the cell functions. What occurs in the nucleus is going to dictate what kinds of proteins and how much proteins and when proteins will be synthesized. If we take a look at this membrane that surrounds the nucleus, it is a selectively permeable double membrane. Within this we see pores, and what's enclosed within this is something called nucleoplasm. This has essential solutes. If we think about nucleoplasm, this is the specific cytoplasm that you would find within the nucleus as opposed to the cytoplasm, which is general to the cell. Taking a look at the nuclear envelope as well, the membrane is going to be studded with ribosomes and is actually continuous with the rough endoplasmic reticulum, which in the previous lecture we had stated was important for the production of proteins. There are these pores that are going to be located within the nuclear envelope, and these are going to help to regulate the transport of larger molecules into and out of the nucleus. Within this, we also see the nucleoli. These are darker staining bodies. This is where we would have ribosome production occurring, the actual production of those ribosomes that will later on make the protein. Now within this, we are going to see that the entirety. Now the important component here within this is going to be chromatin, and chromatin is really a form of DNA. DNA, just to quickly review, it was mentioned in Chapter 2. DNA is a complex molecule made of nucleotides. DNA is a double helix. The nucleotides that are important are adenine, guanine, cytosine, thymine. We have a sugar phosphate backbone creating this double helix structure of DNA. Within the cell, the DNA is stored in chromatin, and these are some different ways to look at the chromatin. Chromatin is actually the thread-like strands of DNA, the molecule itself, associated with proteins called histones. So here in this example we can see the DNA double helix. The rungs of the ladder represent the base pairs. The sides of the ladder are the sugar phosphate backbone. And we can see that this very thin 2 nanometer diameter DNA molecule is wrapped around these histones, which are arranged in units called nucleosomes, and these nucleosomes act like beads on a chain, if you will. These then coil in, and form condensed chromosomes. So you can imagine different ways of looking at this, that within the nucleus, the chromatin exists basically as this fiber, and the fiber is wrapped up DNA helix that is wrapped around histones. Those histones are wrapped around one another to create the chromatin. Now the chromatin is going to condense and is going to form condensed chromosomes when these cells are going to get ready to divide. Now the reason that DNA is important, DNA is going to contain the information that will tell the cell how to make proteins. Now the way this occurs is the 2-step process, and those 2 steps are transcription and translation. Transcription and translation are going to be the ways in which we see the information go from DNA, which is where we have our genetic library that contains the blueprints. That DNA is going to be transcribed through the process of transcription into something called mRNA. Now, mRNA is going to be a portable form of just the area of interest, so just an area that we will call a gene or a transcription unit, which is the information to make a single protein. That mRNA is portable, it can then move outside of the nucleus, and it can then be translated through a process called translation by the ribosome, translated into a protein. And so through that two-step process, we can actually see the creation of protein from our DNA. Now, there are a whole bunch of things that are required for transcription to occur. Transcription, the way this DNA is wrapped around histones and wrapped around in those nucleosomes, the DNA cannot be easily accessed. So before transcription really occurs, we're going to see several transcription factors. And these transcription factors are going to have different roles in terms of loosening the DNA from the histones, binding to a region called a promoter, and that promoter is where on the transcription unit we would see the DNA sequence. The promoter then is the area of, it's a DNA sequence that specifies where we will start transcribing our RNA. So this would be at the beginning of our transcription unit or our gene. So these different transcription factors are then going to help the important enzyme which will actually do the transcription. And that enzyme is called RNA polymerase. When you have something ending in ASE or ACE, that tells you it's an enzyme. And enzymes are often then named by what they do. So this enzyme, RNA polymerase, is an enzyme that makes an RNA polymer, or a chain of RNA. Now, in a simple sense, the way that this occurs, the RNA polymerase is going to help to read the DNA code, right? And then that DNA code is going to be copied, and we're going to be functioning, RNA polymerase is going to function to add the complementary RNA to where the DNA is. So if we take a look at this, here is our initial double-stranded DNA. This is our transcription unit, or what we will refer to as the gene. This is the information to make a single protein. This region here is the promoter region. When this begins, we see the DNA unwinds, and the RNA polymerase will bind to the promoter region. This will all be mediated by transcription factors. As the RNA polymerase moves down along the DNA molecule, it is going to be adding RNA nucleotides to create the mRNA. So as it moves along, we see the beginning of this mRNA. And as it elongates, it actually copies, if you will, this DNA, leading to the production of this mRNA strand. We call this an mRNA transcript. What it is we have two DNA strands, one that's called the coding strand and one that's called the template strand. The mRNA is created off the template strand, so that it is a near exact copy of the coding strand. Now, there are some slight differences between DNA and RNA. One of the key differences is that DNA is always double-stranded, whereas mRNA is single-stranded. Another main difference is that in DNA, the four main bases are A, T, G, and C. However, in mRNA, there are no T, there are no thymine. So wherever there would have been a thymine, there is now instead a uracil. And that is a very important distinction between the DNA and the RNA. Now, there are different kinds of RNA that can be created. The one that we're looking at here with transcription is messenger RNA. And this is actually going to carry the genetic information from the DNA in the nucleus to the ribosomes for the actual translation into protein. That translation, which we'll see momentarily, requires two additional types of rRNA and tRNA. The rRNA, or ribosomal RNA, this is a structural component of the ribosomes, and when we looked at the ribosomes early in the chapter, we mentioned that. This other kind, the tRNA, or transfer RNA, this is going to be an important type of RNA that will actually be bringing in amino acids to allow the protein synthesis to occur. If you remember from chapter 2, proteins are chains of amino acids that have been bound together. So once we've gone from the DNA within the nucleus, we have selected the gene that we are going to transcribe. We have taken that transcription and created our portable mRNA strand. That is then going to leave the nucleus and make its way to the ribosomes, usually on the rough endoplasmic reticulum. At that point, we then see the initiation of translation. Once the mRNA finds its way to the ribosome has two subunits, and we'll see that the mRNA is going to attach to the smaller subunit. The first part in looking at translation is always going to occur when we have something called an initiator tRNA that binds to the small subunit. This initiator tRNA carries with it a very special amino acid called methionine. Right? So we say that it's a methionine charged initiator tRNA. Once that tRNA binds to the small subunit, we now have the large subunit coming into play, and we have a functional ribosome. Okay. Those are really important steps in terms of the translation. What occurs with translation then, if we take a look at this, right? So let's look at this visually. Right? Here we have the nucleus. This would be the entirety of the DNA. This is our gene that we are interested in, so we are making an mRNA. That mRNA is a copy of the coding sequence. It is the complement of the template sequence. This will then get released from the pores. It will make its way over to a ribosome. The first step, not shown here on this diagram, that will occur is the correct codon for tRNA methionine charged initiator will come into play, and that happens to be what's called AUG. Now this ribosome has three main sites on it, the E, the P, and the A site. The E site I like to think of as the exit site, the P site I like to think of as the peptide site, and the A site would be the amino acid site. As this mRNA is getting read, what DNA and mRNA really are, these are triplet codons. So these three letters here, they can be translated into an amino acid. So for example here, our next codon is AUA. The complement that would match that then would be UAU. So here on the mRNA, in the amino acyl or amino acid site, we have this AUA, which is being quote-unquote read by the ribosome. What that means is a tRNA that has the appropriate complement that can fit in will come into place and will bring with it its particular amino acid. In this case, the UAU codes for leucine, which is an amino acid. So this will come into place, the complements will hang out together. This brings an amino acid in. Now previously, if we look at the previous step, here in the P site, this is what we consider the peptide site. So sitting in here, what we have is we have a tRNA. This tRNA has an anti-codon, CGG, which is the complement to the codon of GCC from our mRNA. It has the amino acid alanine, and if you notice beyond this there is a chain of amino acids. This is your growing peptide. Now what will occur here is you can imagine this tRNA will move into place. This leucine and this alanine will be next to one another. A bond can form between these. It releases the tRNA, and then the tRNA, when it no longer has an amino acid, it can be released. What this leads to is as the ribosome works its way down the mRNA, reading these triplet codons at a stretch, it is building the appropriate protein. So this figure that we are looking at right here, this is as this process has already been occurring. Okay? So this is how this has already gone through quite a number of steps. Okay? Reading along. This will continue until a codon here, that's known as a stop codon, is read. And at that point, that stop codon will separate the small and large subunits of the ribosome, and then we'll end this process. Now, the way of understanding this translation is to understand the way the codons work. Okay? This is basically a language. So we call this the genetic code. And within the genetic code, what we see is we have a triplet code. Now, you do not have to memorize this. What you do need to memorize is this shown in green, AUG, which is called the start codon, and it codes for methionine. We previously mentioned that the first codon is a methionine charged initiator tRNA. So when AUG is read on an mRNA, that is the signal to start translation. Now, stopping is one of these three, UAA, UAG, or UGA. And these do not code for an amino acid. The rest of these, we have this repeating series where we have the first base, the second base, and the third base. And what you should notice is that, for example here, we have leucine. This is an amino acid. There are 4 possibilities of what triplet codon would code for leucine. This is very important in terms of how DNA works. DNA is prone to mistakes. We will shortly look at DNA replication. We'll see that mistakes sometimes occur. This helps to prevent changes in amino acid sequences due to changes in DNA. So this is what's called a wobble or a degenerative code. Now, in short, if we kind of give the overview of information transfer from DNA to RNA, the DNA triplets are transcribed into mRNA codons by RNA polymerase. Those codons base pair with the tRNA anticodons at the ribosomes. Those amino acids are going to be peptide bonded to form polypeptide chains, and we have that start and stop codon. Looking at this visually, you can imagine here would be our DNA molecule. We have numbers of different genes. Here we're looking at just a section of it. This would be the DNA sequence. Here we see the transcribed mRNA sequence. This would be the translation step leading to our final protein. In terms of what DNA does and why it is so important, DNA, every cell in your body, contains all of the DNA to make all of the proteins that could possibly be necessary for any of the cells in your body. What is going to separate, say, a liver cell from an eye cell from a hair cell is what proteins are made at what point in time by a specific cell. And that's going to have a lot more to do with development than anything else. When things go wrong with DNA is when we have fairly massive problems occurring. Now, at the very beginning of Chapter 3, we mentioned the cell theory and said that the continuity of life is cellular in nature. We will see that cells divide themselves in order to create new tissue and new cells. So as this division is occurring, we have to understand that the DNA must be divided in a meticulous manner so that the DNA that results in 2 daughter cells or 2 newly created cells is exactly the same. So what we will look at now briefly is an overview of the cell cycle, including mitosis. And this is basically the life cycle, if you will, of a typical cell. Now, all cells have the ability at some point to divide. Some cells cease dividing, but how cells renew themselves and how cells reproduce is through division, specifically mitosis. Now, for the most part, most of the time of a cell's life is going to be in what's called interphase. And interphase is the time in the cell cycle when the cell is not actively dividing. Interphase, though, there is a lot that's going on during interphase. We have the G phase, and this is a growth phase. This is when the cell is growing. It is producing the protein products that are necessary for that particular cell. There is also the S phase, and this is the synthesis phase. During the synthesis phase is when we have DNA replication occurring. This is going to be very important, and we'll look at this briefly. How does DNA synthesize itself? Then we have G2. This is additional growth and getting ready for division. Now, the division occurs during the mitotic phase. We will look at some subdivisions of the mitotic phase. But mitosis, overall, is the process of taking a single cell and reliably dividing the genetic components, dividing the DNA, to create two new daughter cells. This also involves cytokinesis, and cytokinesis is the process of dividing the rest of the cell components, the cytoplasm, the plasma membrane, and the like. Now, in terms of these G phases, there is a lot that goes on. We sometimes see cells that go into what's called a G0, and these are cells that stop dividing. So these are cells that can no longer replicate themselves. Now, the DNA S phase, the DNA replication or the synthesis phase, this is extremely important. This must occur before mitosis occurs. What this is, every piece of DNA within the cell replicates itself or makes a copy of itself, so that when the cells divide, those two copies can be separated, and both of the daughter cells now have all of the DNA that was originally in the cell. That gap 2, or G2, is basically setting up the additional things that are necessary for division to occur. So we'll just take a few minutes and look at DNA replication before we talk about mitosis. Now, DNA replication is very complex. We're doing an overview of it. To remember, you have your DNA double helix. It is made of complementary base pairs where you have A always forming 2 hydrogen bonds of complements with T. G and C form 3 hydrogen bonds, so they are connected. What we are going to do in DNA replication is we're basically going to open up these 2 halves, separate these hydrogen bonds, and create new copies of each of these existing strands. What is going to begin this is the DNA has to unwind from those nucleosomes. Remember, DNA, the molecule, is wrapped around histones in the nucleosome, so it's tightly wrapped around a spool. You can't do much with it. So in order for replication to occur, first we have to see some unwinding from those nucleosomes. Then an enzyme called helicase comes in. It ends in ASE, so we know it is an enzyme, and helicase is going to start untwisting this double helix and start separating out these 2 strands. Okay. Where this opens up, this is called a replication bubble, or an origin, and we're going to see that from this, 2 completely new DNA molecules will be created. So the nucleotide strands, the old nucleoside strands, are each serving as a template for building a new strand. This is different than what we saw in transcription, because in transcription, only the template strand was used to make a copy for the mRNA. Here, each of the 2 strands, we'll call these the 2 old strands, have a new strand made on them, so that the new DNA molecules are half old strand and half new strand. Now, we're going to just look at one strand for this diagram, right, just to understand it. There's a group of enzymes called a replisome. They are enzymes like primase and replicase, and these are going to start this whole process off. And they're going to start this off by using a little piece of RNA called an RNA primer. Okay? And that primer is going to be just enough to get this started. Once this gets started, the molecule, the enzyme that is going to create the new strand, is called DNA polymerase. Previously, we saw RNA polymerase when we were making an RNA strand, but now we're making a DNA strand. So this is DNA polymerase. This DNA polymerase picks up on this RNA primer, works its way down along the DNA template, inserting the complements for whatever base it is reading. So if it sees an A, it puts in a T. If it sees a G, it puts in a C, so on and so forth. Okay. Until it has worked its way down to the next replication bubble. Now, what's interesting is this DNA polymerase can really only work in one direction. Now, we haven't talked too much about the direction of these molecules, But these molecules do have a very particular orientation. If you think back to chapter 2, when we looked at DNA, the DNA have that sugar in their sugar phosphate backbone, and that sugar is a 5-pointed deoxyribose sugar. So you can imagine with that 5-pointed, kind of like a pentagon, the oxygen in it is facing up towards the top. So in one of these strands, they're all in the same direction with that oxygen facing towards the top. In the other strand, they're all upside down. And if you don't remember that, go back to the notes from chapter 2 and look at the DNA section to look at the structure of the molecule. So what that means is this DNA polymerase can only work in one direction. So as these molecules are going along, one strand is going to be copied continuously from the beginning to the end all the way through. The other strand is going to have to be copied in short bursts. So you would have a short burst, and then the molecule would fall off, move up, and work forward. So this what's called the leading strand just works in the forward direction. The lagging strand then takes 3 steps forward, 2 steps back, 3 steps forward, 2 steps back, in order to create the lagging strand. Those little sections are called Okazaki fragments, and they are then bound together or glued together with a molecule, an enzyme called DNA ligase. At the very end of the chromosomes, we have regions called telomeres, and those you can think of as being caps on the chromosomes. And this process altogether is called semi-conservative replication. Semi-conservative replication means that we are somewhat conserving our initial molecule. So each of the 2 new DNA molecules has one old strand and one new strand. One old strand, one new strand. That is semi-conservative replication. Now when this DNA replication occurs, it occurs during S phase. So if we think back to that cell cycle, we have a brand new created cell that has all of the DNA that it needs. It then grows, and when it's getting ready to divide, it copies all of its DNA so that when it divides, the 2 new cells that arise from it can each get a copy of that DNA. So that is really the point of DNA replication. So the process of dividing then is called mitosis. And we're going to run through mitosis in an overview form. Mitosis has several different sub-phases. Depending on what book you're reading and what source you're using, you can get progressively smaller and smaller and smaller in these. The main sections are usually prophase, metaphase, anaphase, telophase. Sometimes you will hear things referred to as early and late prophase, or pro-metaphase being a phase between prophase and metaphase. Right? These are just different ways of looking at subdivisions. Now in early and late prophase, here's where we're going to start seeing those centrioles that we mentioned before. Those centrioles are going to start moving towards opposite ends of the cells. The nucleoli, which produce the ribosomal RNA, are going to start disappearing because we don't need ribosomes right now. When a cell is dividing, it's not making a lot of other proteins. Now we start to see what's called an aster. That aster looks like a star, and this is due to those 2 centrioles releasing fibers that are called the mitotic spindle. And these are going to start moving away. Now as we get later in prophase, we'll see the asters are more completely towards opposite sides of the cell. The DNA is now in very clear condensed chromosomes. Those chromosomes are no longer held within the nucleus. In fact, the nuclear envelope has broken down. Each of these chromosomes are actually copies held together. So the initial chromosome and the copy that was made during DNA replication are now being held together. And they're actually hooking onto those spindles that have been created by the centrioles. And we'll see they're going to start moving back and forth. In metaphase, those chromosomes will all line up along what's called the metaphase plate, or the equator of the cell. The centrioles are at the opposite poles of the cells. The spindles that go towards the middle are actually what are going to push and pull and get these chromosomes lined up on what's called the metaphase plate. In anaphase, the centromeres of those chromosomes are going to split. So we're now separating the copies of the different chromosomes, and these are going to move away from one another. So now we see these chromosomes moving towards this pole, and the other copy of each of those moving towards the other pole. The way these are moving is that motor proteins are going to actually pull the chromosomes up along the spindles. Now in telophase, and then finally cytokinesis, in telophase we start to see the reappearance of the nucleoli. We start to see the chromosomes unwinding. We start to see the reforming of the nuclear envelope. We will also see cytokinesis occurring. Cytokinesis is the division of the rest of the cell components, and this usually occurs when we see what's called a cleavage furrow. And this is, imagine a pinching off between the two cells, and that will lead then to the final separation and creation of two new cells. Now the way I usually remember this is in between mitotic divisions we have interphase, then we have prophase, metaphase, anaphase, and telophase. I often try to use mnemonic devices to remember things in order. So I remember I put my act together. I, interphase, is in between. P, prophase, is getting prepped. M, metaphase, is getting in the middle. A, anaphase, is moving away. And T, telophase, we now have two new cells. Whether or not you find this helpful is really up to you. Just a final suggestion.